No? Okay, great. So we're all familiar. We're the largest remaining natural area here in Pinellas County. We protect our native flora and fauna. And we're also an education center here. If anyone's never been, you should go check it out after today's program. We are constantly doing programs like this one today. We're run by Pinellas County and UF IFAS Extension. If you're not familiar with extension, basically it just means that we bring research-based education to the public like we're doing today. And we're always doing guided hikes, programs like this, festivals like last month we had Owl Week and Wildfire Festival. And so we're happy you're here. And today's program is graciously funded by the Friends of Brooker Creek Preserve. So if you're interested in learning more about them, we have pamphlets at the front. And if you have any other questions, you can come to me, Julia, about anything Brooker Creek related. And so today, I want to introduce Jenna, and she's from UCF, and she's coming here to present her research on the lung parasite that could possibly be contributed to the decline in snakes here at Brooker Creek, and just in general. So, we'll welcome her up.
Thank you for the whistle. That reminded me of my dad. So thanks for coming out. Again, I'm Jenna Palmisano. I'm a second-year PhD student at the University of Central Florida, and I'm in Dr. Anna Savage's lab. So she mostly studies amphibians, so I'm the new reptile gal in that lab. So today I'm going to be talking a lot about pygmy rattlesnakes, so we'll start with an introduction to those cool guys, and then we'll talk a little bit about what's contributing to their decline across Florida. So just a little outline. I'm not sure how familiar you are with the life history of Sistrurus miliarius barbari, the dusky pygmy rattlesnake. So I'll do a little bit of an introduction into them, what their behaviors look like in Florida, and what their populations look like too. So, and then we'll talk about the threats to dusky rattlesnakes here in Florida and the emerging infectious diseases of most concern. So that's what EID stands for. We're mostly going to be looking at snake fungal disease and pentastomiasis. So there'll be good background for both of those diseases, so no worries if you don't know what they are. We'll also look at some evidence of population declines in pygmies. And then we'll talk a little bit about my dissertation studies and the progress that I've made thus far. Again, I'm just a second year, so not too much data to work with, but I've got about 200 snakes since June. So we'll talk a little bit more about that. But if you don't know much about pygmy rattlesnakes, pygmies are vipers, so they're part of Viperidae, which is divided into two subfamilies. So pygmies are pit vipers, so they do have heat-sensing pits. And there's 3 subspecies across the entire range of pygmy rattlesnakes. Here in Florida, we just have 1, so that's Sistrurus miliarius barbari. And then there's the Carolina pygmy rattlesnake and the western pygmy rattlesnake. So they're found kind of throughout the southern quarter of the US. And like most pit vipers, they are ambush predators. So you can see a pygmy here kind of in the indentation of a fallen palm, which is very cute to walk up on. They're definitely really cute, and they always look super comfortable and cuddly, even though they're not. So about their life history, they typically reproduce around 3 or 4 years old. That's when they start, but they can start as young as 2 years old. And usually reproductive individuals are between 50 and 150 grams. The average of like the 200 snakes that I've caught have been around 50 to 60 grams, with some monsters actually reaching up to 175 grams. So you'll see a picture of the Pygmy at the end of this, just to end on a good note. But they're usually quite small, under 2 feet. And in Central Florida, the dusky pygmy rattlesnake has really only been studied in one population, and that's more on the east side in De Leon Springs. And that population has been studied since 1992. And that's kind of where all of our life history data about the dusky pygmy rattlesnake comes from. So there could be variation throughout the peninsula and in the panhandle. I'm hoping to elucidate some of that with my studies, but we will see. So our litter sizes in Florida are usually between 2 and 12, with an average of, I think, about 6. And then that's typical for the western and the eastern pygmies as well. And these guys are usually eating anurans, so frogs and lizards as well, and sometimes they eat centipedes too. So like all species, one of their biggest threats is habitat loss and degradation. This is just an example of habitat loss with our historic range of longleaf pine. Now we have less than 2% of that continuous range, unfortunately. What do you need?
You're good.
Okay, cool. So again, more threats to Sistrurus miliarius barbari is roadkill. So can you guys find the pygmy on this road? Anyone see it?
Is he in the center?
He's kind of in the center. He's a little bit off. He's right here. So you can see why hunters, if they're not looking for snakes, can easily mistake a young pygmy for a twig. So they get hit a lot on the roads, especially on these off-roads. So this is a picture from Osceola. Also invasive species prey upon them, so hogs do a lot of damage to their habitat and either kill pygmies by eating them or stomping on them. And then unfortunately, this canebrake rattlesnake is an example of persecution. Some people still do persecute pygmy rattlesnakes, unfortunately, so that's another impact to their populations. But today we're mostly going to be focusing on a threat on top of threats, the emerging infectious diseases that are most prevalent in dusky pygmy rattlesnakes. So emerging infectious diseases are increasingly recognized as contributing factors to wildlife population declines. So some pathogens can cause mass mortality and morbidity, and some others, like avian influenza, get a little bit more attention because there's zoonotic concerns, which means the disease can infect humans. So we'll start with snake fungal disease. Snake fungal disease in Florida and pygmy populations, it's been around since the first detection was like in 1990, in the early 1990s, so it's been around for a while. On the left is more severe cases that we've seen in the past where there's kind of swelling around the facial pits, and also that's like typical, this brown crust. You can see it on the ventral scales here as well. So these diseases can become systemic when they get into the body cavity, but often with pygmies we don't really see a lot of facial lesions. We see a lot on the back end of the animal, lesions around the cloaca, around the tail. Sometimes it will cause the actual tail to fall off, unfortunately. And snake fungal disease is really widespread. It's across the globe, so I mean mostly in North America, where it's highlighted here, and then in Europe as well. So it is keratinophilic, so it's feeding on the keratin of the snake scales, but then it does send hyphae into the body cavity, and that's when the disease can become systemic. And the causative, or the primary causative agent of this disease is Ophidiomyces ophidicola. And it impacts over 60 snake species globally, but you can see there's kind of a hot spot in the eastern US, and that's where we see the most severe symptoms as well, as you can see in these pictures too. So definitely disrupting these animals' feeding. But again, in pygmies, we often don't see a lot of facial swelling. We see a lot more of it on the posterior third of the body. But that doesn't mean it's not causing an issue. Just a little bit more background on snake fungal disease. Again, it was present in Central Florida at least since 1992. We have evidence of it in the US in 1940s, but we do believe that it is a introduced and exotic pathogen. So it's widespread in Florida. We've found it in the Panhandle all the way down to the Everglades. But the unknowns in Florida right now are kind of the strain diversity and how those differences in strains relate to the severity of disease, and whether or not the scoring systems that we have available to us actually indicate the outcome of the disease. So there's 3 different types of scoring mechanisms, and they all relate to like the type of lesion that you're seeing, whether it's an ulcer or just a discolored scale or a raised scale. Also takes into account how big the lesions are, as well as where they are on the body. So I kind of use lesions ubiquitously, so like I would call all of these lesions, but that's not how the range works. And like when I'm taking data and stuff, I won't call this a lesion. I just call it my lesion data sheet, and then I decipher which ones are actually considered lesions versus like an ulcer. I've only seen one ulcer and it wasn't that bad, but often you can see just displaced scales, thickened scales, scabs. But a lesion itself is kind of an open wound. So there has been some genomic work done to separate different strains of snake fungal disease into 3 different clades, but additional work again needs to be done in Florida to really understand the correlation between strain presence and the severity of disease. But in pygmies, what we're seeing are quite a bit of sublethal impacts, and these are probably exhibited in other snake species as well. So there's a febrile response or a fever response. So there's lots of basking. They're basking for longer periods. They're getting to higher temperatures, and they can be a little bit conspicuous, more so than if they aren't sick. So in this graph here, we can see the metabolic rate of these animals. So these are all at different fever regimes. And so these are colleagues of mine who've done respirometry trials with pygmy rattlesnakes that have snake fungal disease, basically putting them in a little jar and calculating how much CO2 they output. And for fever responses, at the time of this paper, we were initiating those by keeping them at those temperatures, and we called those ecologically relevant temperatures from what we knew of snakes' fever responses from taking their coacal temperatures in the field. We now have better data for this, and we actually have empirical temperatures from these snakes because we put tiny little data loggers in them that took their temperature for a few weeks every 10 minutes. So soon we will see this graph with actual fever responses. But what you can see is that snakes that don't have fungus, their resting metabolic date, these open white circles here, is a lot lower than all of these snakes that do have the fungus. And the black one here has no fever response, and the other ones had induced fever responses of 2, 4, and then 5.5 degrees. So what increasing the resting metabolic rate means is that these animals are using energy to combat this disease, whether that be, the common thought is ectotherms are going to raise their temperature so that their immune system can work better, but we don't know if that is what the pygmies are actually doing or if they're trying to reach a temperature that the fungus can't live in. So that's one of the things that we're trying to tease apart now with our research. But some other things, you saw it in those other pictures, you can see lots of inflammation around the lesions, increased stress response, so that was calculated with cortisol levels, and then also altered reproductive cycles. So males and females aren't making the right amount of hormones during the times that they should. So some other sublethal impacts are dysectisis, which basically means it interrupts their shedding cycles, so they might shed a lot more to try to get rid of the fungus. They're typically emaciated, so they get really skinny, and you can feel that with kind of spinal definition by just squeezing them a little bit. Anorexia, so they're not really eating, they're basking a lot more and spending a lot more energy on their immune system. And it also increases their evaporative water loss. So we do see symptoms of dehydration, which kind of just looks like a really baggy snake. And if you pinch their sides, the skin doesn't go right back. It just kind of stays. And so this is just a graph of evaporative water loss. And so you can see these snakes that were negative for SFD in the open circles here have less evaporative water loss than the infected snakes. All this to say is that snake fungal disease, although it may not cause mass mortality, it is adding an energetic cost to pygmy rattlesnakes. And whether or not that impacts how many babies they make in the future, that's something we're trying to figure out. And then on top of that, how are the impacts of a single pathogen? How do those interact? What are the dynamics with co-infections of other pathogens that we see in pygmy rattlesnakes? Any questions about snake fungal disease before we move into pentastomes? Yeah?
Can it be treated, or can the fungus be treated proactively?
By the snake naturally, or more like us intervening?
Could we intervene and treat the fungus?
Yeah, I don't know of antifungal treatments that work for SFD. I'm trying to think if there's been any studies, like there's gotta be. But not that I know of right now. I can't think off the top of my head. But I know like in other species that deal, like in amphibians that deal with chytridiomycosis, there are some things that treat that fungus. So I just don't know. I can't think off the top of my head. Maybe. And a lot of times we do see snakes clear infection with like numerous sheds in the warm, dry months. So like coming out of summer, snakes should look really good with plenty of food in their bellies and plenty of hot weather so that they could reach temperatures where their immune system is working really well, and they're also able to kind of induce plenty of shed cycles, and they can clear it. And basically what that means is that if we swab it and then extract the DNA from that swab and there's no fungus on it, then we're saying it clears it. But do we know maybe if that snake might still have a systemic bodily infection? I'm not sure, but we do see snakes look better after like the spring months and stuff. So sometimes they can clear it on their own, but I definitely need to look into any type of antifungal agents that people have used in captivity. I can't think of any right now. And I know there's some researchers that are looking at microbiomes of snakes as well to see if any of the natural bacteria that live on them are helpful in any way to this disease. You had a question?
Yes. Do we know any vectors? How does the snakes get this?
Yeah, so we don't. The thought right now is that it's just coming into contact with soil that has it, or other snakes that have it. Yeah. Lots of unknowns. Yes.
So I've run across a few pygmy rattlesnakes lately. They haven't been rattling at all. And I don't know if maybe you said they could lose their tail.
Yeah.
And does the lethargicness cause them to maybe not want to move as much, not want to rattle?
Yeah.
Because generally it isn't a basking situation where they're just not making any noise, they're not moving.
Yeah.
When provoked to get off of a trail, to move them out of the way, they're not wanting to move, so.
Yeah, that definitely could be the case. They might also be in a tight ambush coil. Usually if it's like a looser coil on the ground, then they're thermoregulating, and if they don't move out of that, then I would be like, that's concerning. But if they're in like a tight little ambush coil, sometimes what I've seen with a lot of different rattlesnakes is that they just stay in that tight coil. You can pick them up with tongs and they'll just be like a little cookie, and they don't want to move because they're like, you don't see me, it's fine. So I think it depends. And then also another thing with pygmies is, for unknown reasons, they lose their rattle a lot. I think I most commonly see, 2 or 3 segments, and then there's kind of like the anomaly of snakes that have like 11 segments or 7 segments, but often you don't see them and often you don't hear them. But seeing lethargic snakes is definitely concerning. I would say that's usually a symptom of some type of disease. Could be any of them. I definitely see lethargic snakes that have pentastomes, which we're about to get into. But
They're not in their tight coils. They're usually almost pancaked out. In the sand.
Yeah, that loose coil is usually indicative of thermoregulation. And if it's like a really hot day and a snake's doing that, they're typically trying to like cool off and regulate their temperature, but I would have to know like case by case if I could be like, we do usually see loose coils with snakes that are sick, more so than snakes that aren't. Does that kind of roundabout answer your question of like, basically, I don't know the answer to your question? It's like, here's some evidence that could point towards that, but I can't say it definitively. Oh, yes.
I have one more.
Yeah, of course.
Have you personally gotten to the point where you carry a spray bottle of isopropyl and spray off your hook every time?
Oh, yeah.
Okay.
Yeah.
Would you suggest that herpers of all kinds do that?
Yeah. Yeah, I definitely would. I think we don't really know much about the spread.
I've
Yeah.
never seen a herper do that, and now I'm going to start yelling at people.
Yeah, I mean, 70% isopropyl will definitely, it will kill the fungus. It won't get rid of the DNA. So when I put pygmies in tubes to sample them, like I'm always using isopropyl on my tongs, but with my tubes, I usually bleach these and then rinse them off really well with water, just so that if I swab another snake and I get the DNA of the fungus on it, I'm not getting false positives. But yeah, so isopropyl, I isopropyl my boots before I walk around a new place. I bleach everything in that kit between sites. Is it overkill? Probably. But yeah, right? It's better safe than sorry. Yep.
Should we, if we are finding these snakes healthy or not, should we be reporting to FWC so that they're able to get a better idea of like breaking if they're healthy and unhealthy or something else?
Yeah, I mean, with pygmies, I'd be happy. I have my email at the end if you want to chat with me. But I just don't really think a lot of people are looking at the abundance of snakes. So unless it's like a really protected species, like an indigo or a pine snake, I would definitely call FWC. And I have a couple contacts that you should definitely reach out to specifically. But if it's other snakes, and you're not in like a secret location, I think iNaturalist is a good place to post it. I get nervous with that because of poaching. So I typically don't post vipers. So it's a kind of a tricky thing, but I mean, I record all snakes that I see all the time. Will someone ever use that data? I don't know. Yeah, so I don't have a great answer to your question. There's definitely like the Orion Society and the Rattlesnake Conservancy, they want to know every eastern diamondback that's found because we're trying to put them up for protection. So yeah, it really just depends. Yeah.
So any snake
Yeah.
can get this snake fungal disease.
From what we know right now, yeah, most are susceptible. I can't think of one that's not susceptible.
So do you only want to know about pygmies with it, or do you want to know about any snake that seems to be strange?
So I will talk more about what I can do if snakes seem strange on the pentastome side of things. With SFD, I could point you who to talk to with certain species that are studying that. But I only study snake fungal disease in pygmies. But yeah, hopefully that answers your question maybe. If you see 6 snakes, there's a page at the end. I'm actually a part of Partners in Amphibian and Reptile Conservation, and I'm one of the leads of HDAS, which is our herp disease alert system. So if you see a sick herp, whether it be an amphibian, a reptile, snake, whatever, you can report it to us, and then we'll get in touch with the right state representative who can do something about it. So there's a QR code for if you see any sick reptiles or amphibians. Yeah, did you have a question? Yeah?
Yeah, just, I'm not a snake specialist.
Yeah.
How often do they normally shed? How many times a year?
It
And then when they shed, because they're trying to get rid of this fungus, does that shedding contain the fungus for a long time, and can it affect any other creatures? Does some other creature use that shed material?
That's an excellent question, and unfortunately I don't know, and no one has really looked at it, but the fungus, you can definitely like culture the fungus in a petri dish from a shed of an infected animal, and you can see those lesions. The DNA is there, the fungus is still alive on the shed. How long it lives, I'm not sure. Can it be transferred to like a bird that uses it in its nest? I'm not sure. Yeah, and it does increase their shedding, but I can't think of the actual, like, any papers that study the, like, actual increase of it from their normal. And it depends on the species, and it depends on how fast they're growing, so how young they are, of when they're going to shed. So, like, little snakes are going to shed way more often because they're growing faster than larger snakes.
When they shed, do they have to utilize like a hedge or something to make it come off?
Yeah, so sometimes they usually, yeah, some type of branch or something that they can just kind of like get it on their neck, and then they'll kind of go on throughout there. And like some of the issues is that you'll get a lot of stuck shed with snake fungal disease. Like I had one animal last weekend that just had like layers of stuck shed, and just kind of like sores and ulcers underneath that stuck shed. So sometimes if it's not like one fluid shed, it can make things worse. Yeah. Yeah, of course. All right, we're gonna move on to pentastomes. If there's any other questions about SFD, I can talk at the end. But this is the lungworm that we're going to talk about for the rest of this presentation. And pentastomiasis is the disease that are caused by these lungworms, and the one that we'll focus on is the one that's infecting pygmies, and that's Raillietiella orientalis. So you can see it in a Burmese python's lung here, and then this is just a picture that I took under a microscope. So you can kind of see what the head looks like. It's a little triangular-like, with those posterior hooks being larger than the front ones. So they look a lot different than the native species of lungworm that infect some snakes. So just to give you an overview of who pentastomes are, they're part of a class of crustaceans that are known as pentastomes, and their closest relative are fish lice, which you can see on the left here, which are kind of cute. But these are worm-like crustaceans, and they typically reside in the upper respiratory tract of carnivorous vertebrates, and those are most often reptiles, but they do infect, some species infect mammals, so some actually live in the nasal cavities of lions, for instance. But what they do when they live in the lungs or any other part of the upper respiratory tract is they're feeding on the blood, and that's where they're going to reproduce, and the eggs are typically passed with the feces. And on the right is just kind of an example of their differences in morphology. So there's 9 different types of pentastomes here. The one that we're talking about today is not on there. And then vermiform, that just means worm-like. That's why we call them lungworms, but they're actually crustaceans. And this is what all 3 of their life cycles look like. So they have embryonated eggs, which you can see in snake feces, and then they infect an intermediate host or 2, and they look like this. And then they, when they're adults, they're much longer and more worm-like. So typically in snake feces, you can see 2 membranes, but sometimes there's only 1 because you've destroyed that beginning one. But they'll have these hooks, these 2 paired hooks or 4 paired hooks throughout their life cycles. And some of the pentastomes will have an arrangement that's more like this, with all hooks being the same size, but the ones we'll talk about today, they have very interesting hooks. So all of them are going to pass through those 3 life stages, and we'll see why the invasive one's a little bit different than the native. But these 2 are kind of the most prevalent in our native snakes, and these are native to North America. So there's Chirocephalus coarctatus. The life cycle on the right here is for Porocephalus crotali. But Chirocephalus coarctatus utilizes fish as intermediate hosts, so they're most often infecting water snakes. And then Porocephalus crotali is most often using mammals as intermediate hosts, so they're most often infecting vipers, pythons, and large colubrids. So the reason why I put this number here, then this is just from what we know. A lot of people don't study these, so this is what we understand from the literature right now. But 27% of all North American pit vipers have been documented with Porocephalus crotali. And the reason why this matters to me is that those 27% might have some coevolutionary history with these parasites, and therefore might have a better chance with the invasive parasite compared to the vipers that don't. And unfortunately, pygmy rattlesnakes have no evolutionary history with pentastomes. So, like I was saying, surveillance has really been opportunistic, so we don't know much about what the prevalence looks like. I find them quite often, Porocephalus crotali, in cottonmouths, which you can see here, and that's a Porocephalus crotali. I also find them in eastern diamondbacks, but it's typically only like 2 or 3. But we really don't understand if these parasites are pathogenic or causing disease, or if they're just kind of there. But they probably do have some type of sublethal impact we don't understand at this time, and what that means when it comes to being infected with the native and the invasive, we just don't know. So we know even less about the invasive, which is called Raillietiella orientalis. And I'll refer to it as RO because it's a bit easier. But what we do know is that it was likely initially spilt over with the Burmese pythons in the Everglades. And it causes really severe infections in native snakes. In the Burms, we usually only see a handful of pentastomes, and they're a lot smaller than they are in native snakes. And you can kind of see a variety of infections here in a scarlet king snake, black racer, and then it coming out of the glottis of a coral snake. So the infections do get quite severe. And we'll talk more about the life cycle in a couple of slides, but it's likely that the Burmese pythons are not the only culprit of spillover of this parasite, because they also move in cockroaches and small mammals and small lizards and small frogs, which are also introduced to Florida all the time through different ports and other things. And then those frogs and cockroaches might move with horticulture from Miami up the coast. So it's not likely just the Burmese python's fault. So why is it a concern? It's because it's related to mortality of a few individuals, and it also has been documented to cause lesions of the lung, pneumonia, emaciation, and even sepsis. You'll see the infections do get really severe. And on top of the diseases and other threats that they're already facing, it's really important to understand on top of that what pentastomes, what kind of threat they're presenting here. And we see a lot of variation in the mortality, but in pygmies it seems to be really bad. In last year we found 6 dead pygmies in habitat, all 6 of which had pentastome parasites. And a funny story about this racer, well maybe not so funny, but this racer is actually in really good condition. I found it in my driveway, which is just odd, about 2 years ago, and it had them crawling out of the mouth. So typically when a snake dies and it has this invasive parasite, they tend to move into the oral cavity and try to get out. Our best guess right now is that they're just responding to the lack of oxygen. Are they trying to get into a hawk or another host? I'm not sure. But this is one of the signs of a disease. Of this disease, if you find roadkill and there's worms crawling out, definitely contact me. So more about the potential impacts of RO. Obviously, activation of the immune system is expensive for these animals. And then, like I was saying, lesions, pneumonia, anemia because they're feeding on the blood, and sepsis with really bad infections. And you can see some of the hooks here zoomed in. And this was one of the first infections in pygmy rattlesnakes that we found at our study sites. And this is kind of a gross video of a snake that I had to euthanize that had over 60 of these in the lungs. So this is a really good and bad example of how severe the infections can get. So these animals were still alive. Oh, there we go. And you can kind of just see the peristalsis of their gut, all that brown stuff moving. But these large ones are mostly females. I don't think there's a male in there. Maybe that right here might be a little tiny male. The females are the one that are really big, and their whole body is pretty much just one giant ovary. And they live for a really long time in a snake. That's another thing that can be a cause for concern. So when a snake eats an infected prey item, the adults do not start reproducing until about 80 days. So it takes a while to initiate an adult infection. These are some more severe infections. The 2 on the left are from banded water snakes, and the one on the right is from black racer. They look all shriveled up because they were preserved in ethanol. They look a little bit different. I actually have one that we can start passing around if you guys want a closer look.
Yes, I do.
So this one is a female, and they typically get bleached when they're in ethanol like that for a while. But now I'm just kind of going to go more into RO in Florida and what we know about it. And then we'll start looking at pygmy declines, and we'll wrap up with a little bit of the work that I've been doing. So RO was first found in North America in Florida. And it was first recognized in 13 different species. Dr. Melissa Miller did a lot of that initial work. As you can see, her purple mapping here were the initial spots. And we thought maybe it's restricted to where the Burmese pythons are and just outside of their range. But then we started to find them in Alachua and in Volusia County. And then we found even more evidence of them moving outside of the Burmese python range. So here in the red is kind of the core population of pythons, and then all these yellow squares are infected snakes. So you can see Melissa Miller did a ton of work all the way up the central Florida, in the Panhandle, and then into Alabama. And then the black snakes are snakes that were not infected. So seeing all of this as an undergrad, I really wanted to understand how these pentastomes were moving through the landscape. How were they getting outside of the Burmese pythons and then thriving well enough to make it up the peninsula? So I conducted experimental infections as an undergraduate at Stetson University. We found out that RO likely exhibits a 3-host life cycle. And we'll walk through the steps here, but just a simple diagram to show you. A coprophagous invertebrate, one that eats feces like a cockroach, is going to feed on an infected snake's feces, get infected with initial larvae, and then become infectious to lizards or frogs or toads that eat it, and then those are going to infect the snake. There's a question mark next to the mammal there because we know that it plays some type of role in the life cycle, we just don't know what that is at the moment. But we have seen eastern diamondbacks with infections, which are only going to be eating endotherms. And then, of course, Burmese pythons eat a lot of endotherms too. But since we're seeing specialists, mammal specialists get infected, we know that they play a role. And we do know that one Kenyan shrew has been documented in the wild with larvae. So we don't know much about its role, but we know that they're playing a role. So that's these are the pictures of what the eggs look like. You can see one that has the full membrane here, so they kind of have two, and they do often kind of have that like pink sheen. And where it looks like cracked glass, those are actually their hooks. So not the best pictures, but that's going to be infecting things like cockroaches, and we infected 3 different species. This is what the larvae look like in the cockroaches. They kind of look like tardigrades, but again, we're always having throughout the life cycle those 4 paired hooks, and then that is their feeding apparatus. And then just again, some other examples. They're just in the fat bodies of cockroaches from our experiments. And then when they infect a lizard or a frog, this is what it looks like. So all those white dots outside or right against the abdominal wall here, those are all larvae, and they get kind of double in size from the cockroach to the second intermediate host. So one thing that's different about RO and what probably helps it invade is that it has this 3-host complex life cycle that includes many invasives here in Florida, unfortunately. And other pentastomes typically only have a 2-host life cycle. So there's kind of an interesting evolution when it comes to this species in particular. So these are the records of intermediate hosts that we've found in the wild. Most of this work has been done by myself and Dr. Terry Farrell. So none of this is published, but it soon will be, and you can see that the ones that are highlighted here are 2 of the invasive species that move all over the southeast. So more pathways for RO to invade. So these are the 7 that we know of now. We kind of do this opportunistically because we don't like to kill native lizards and anurans. But there's probably a lot more intermediate hosts than we know of. And so for snake hosts in Florida, only 14 are published. So you'll see some bold text for unpublished findings, but there's 18 snakes that are native to Florida in total that have been found with infections, with the latest one being a mud snake by myself and colleagues at Florida Gulf Coast University. So we had 2 mud snakes that had pretty light infections, but infections nonetheless. And you can kind of see that this is pretty widespread throughout the colubrids, including Drymarchon, which is a protected species, the indigo snake. We've also seen it in several elapids, in several coral snakes. We only have 1 elapid here in Florida. And we've seen it in Sistrurus, Crotalus, and Echistrodon in Florida. So we have not seen it in canebrakes yet, or copperheads. And canebrakes are really only in North Florida, and copperheads are really only in the peninsula, in the panhandle. So the reason why we're concerned with this kind of widespread infection of snakes is that a lot of our snakes are already not doing well, like Heterodon. And Heterodon are frog and toad specialists, and that is the most frequently used intermediate host from what we understand of RO dynamics at the moment. So we're really nervous about that. I've only gotten a couple of samples of Heterodon, so really not a good sample size right now, but we haven't found any with infection yet. So the biggest risk factors are diet, and a lot of snakes either, at least ontogenetically, so their shift from when they're babies to when they're adults, are at some point eating ectotherms. So we're really concerned for the snakes that are already exhibiting declines, like our pine snakes, our indigo snakes, and Heterodon, our hognoses, especially because it uses so many different intermediate hosts and is able to utilize so many snakes, if not all. So detection for intermediate hosts is a little bit unfortunate at the moment. We only know how to detect infections in intermediate hosts with larvae with dissections. In the definitive host, you can also use dissection, so we utilize roadkill or opportunistic collection to find pentastomes, and then you can also find them in fecal samples. So you can see the eggs in their fecal sample, and then I've been able to extract the DNA from the eggs and amplify it to detect that it is the invasive. And then I've started this monitoring group that we call SLAM, which is Snake Lungworm Alliance and Monitoring. And this consists of about 50 to 60 people across the southeastern United States. And we are opportunistically collecting road-killed snakes, and they give them to me and I dissect them and figure out if they're infected. And what we've started to do is work with other colleagues to test for other pathogens as well. So we're not just looking for the pentastome parasite. And then we take a bunch of samples out from that snake, put it in ethanol, then we fix it in formalin and send it off to a museum so that it can be properly stored. But since we've increased surveillance looking for RO, we've actually found 9, I guess today 10, unfortunately, 10 new counties of infections. So unfortunately, Pinellas County needs to be marked off as red. Before I get into kind of what we're seeing with declines in pygmies and how it's related to RO, this is another pathway where RO can spread, and that's the pet trade. So I have found 3 infections in the pet trade. They were all from the same breeder or collector in Miami, and they were sold to Guantanamo Matanzas National Estuarine Reserve, where I have a couple of colleagues that are rangers there. And they started to see their snakes be lethargic and then anorexic, and then one actually threw up some of the pentastome eggs, and then all 3 died within a couple months of being sold. And so you can see 2 of them here. There was 2 garter snakes and there was 1 scarlet king snake, all of which had pretty heavy infections. And another incidence of RO infection in the pet trade was a banded water snake in Michigan. So that's the only place that we know outside of Florida that RO has been in North America. But again, that was a captive infection. And I forgot to note that RO has also invaded Australia. And a lot of that data comes from Crystal Kelly here, and she did a lot of studies around 2014, but no one really studies it there anymore, and it kind of mimics similar patterns as it does in Florida, where it's widespread and the highest prevalence is in snakes that are mostly eating ectotherms. I won't go too far into this, but if you have questions about the phylogenetics of this parasite, we can chat more about it. But basically, as we are kind of playing catch up with this invasion, we need more molecular tools to be able to detect infection and maybe figure out different ways to potentially treat it. And also what it looks like in its new range. How is this parasite evolving within new hosts and in a new landscape? So if you have questions about that, we can talk about it afterwards, but I won't bore you with that in the meantime. So moving into our declines that we're seeing in Sistrurus miliarius. In several different populations, including the Everglades, we're seeing declines aligned with the first detections of RO. And this is just an example of 4 large females that we pulled out of a pygmy rattlesnake from a central Florida population. So this is actually one of the 6 that died last year. This is some data. Really the highlighted column is the most important, but this is looking at road surveys in the Everglades over time. So Dalrymple did a lot of work. As you can see, he caught like almost 3,000 snakes in the 80s and 90s, and he found that about 11% of them were pygmy rattlesnakes. And then in subsequent years, you can see that the percent of pygmy rattlesnakes caught drastically declines to less than 1%. A lot of this data, again, is unpublished, but it also correlates to findings in iNaturalist. And I'm writing a manuscript on this now, so hopefully there'll be more insights, but we've also seen declines at a couple of our central Florida sites that have been studied long term, where we used to go out there, see 30 rattlesnakes really easily, and now I walk like 6 miles there, and it's really hard just to find one. And those crashes aligned with the introduction of RO, or at least the first detection of it. So it's likely, given that Burmese pythons established in the early 2000s, that this is when RO started to get into pygmy populations. So this again might be some boring stuff, so I can just go fast, but this is just some models that I've created based on the oxygen consumption of the actual parasite. And so you can just kind of see the energy of uninfected snakes is higher. But this graph here, it is only modeling the energy consumption of the parasite, not the energy expended by the snake. So that's something that we want to add to this model to get a realistic view of what the energy tax is on snakes with pentastomes. So from all of this, we know that RO is a major conservation concern because it uses widespread taxa as both intermediate and definitive hosts. And that's going to allow it to have an extremely rapid geographic expansion and continue that. And there's not really any known limitations. We've dried out the eggs for 16 plus days and they're still viable and can infect cockroaches. We've infected several different species of cockroaches, two of which are invasive here in Florida. We do know that freezing the eggs kill them, but if they're in an intermediate host, they're not likely to freeze. There are pygmy declines, at least from our data, that are aligned with first detections of RO. And there's some evidence in other species too, but that's not my data, so I can't speak on that part. And again, those 6 dead individuals we found last winter with infections, and we just don't know enough about this disease to know how much of a threat it is. So with all of that behind us, we're just going to talk a little bit about what my studies are, and now you kind of get the background of why I'm doing them. Did you ask me before if you can see if they're pregnant really easily?
Julia did, but
This one's pregnant. Yeah, Yeah, so but that's its tail. So you can see how distended the back end gets. And that was in June. Actually, that one is at Chassahowitzka. So typically, you see a lot of females in June basking and in ambush a lot more than any other individuals because they're trying to get more energy for their babies. And you can typically palpate, it feels like a grape. So like when you slide your thumb, it kind of, each ova slides underneath. But yeah, so for my dissertation, given all the background information I just gave you on snake fungal disease and pygmy declines and pentastomiasis, I'm studying 13 locations, and I'm doing epizootic studies where I'm trying to figure out which populations have Ophidiomyces ophidicola, so the causative agent of SFD, and Raillietiella orientalis, the causative agent of pentastomiasis. So I'm looking if they're there, so I'm looking at prevalence, and I'm also looking at severity of disease. And that's pretty easy with SFD, but with RO, it's a lot harder. We don't really know how to look for severity of disease with RO, so we're working on that. And then the genomic studies that I'll be doing with pygmies, I will be looking at a neutral data set, so kind of looking at genes across their entire genome and looking at what the architecture of their genetic populations in Florida looks like. And then I'll also be looking and comparing that neutral diversity to adaptive diversity with MHC class 2 alleles. So these are major histocompatibility alleles, which pretty much help your immune system flag down antigens. And so I'm looking at if there are advantageous immune alleles within these populations and if that correlates to the disease severity that we're seeing. So if you have questions about that, let me know. We can chat more about genetics. But one of the things within those 13 populations, I'm studying 3 really heavily. And these are my mark recapture sites. And so I go out to these every month, and I'll be doing this for the next 4 years. And I want to assess how snake fungal disease and RO are impacting their growth rates and survival at these sites. And unfortunately, I already have 2 deaths, which is sad. But all 3 of these sites are very interesting because they have different detection times of RO. So they have varied dates of when RO likely arrived there. And they also have different prevalences, with one site having really high prevalence. That's the one that has exhibited decline, extreme decline. One that has kind of a medium level prevalence, and then one that has a low prevalence. So 3 good populations to kind of compare growth rates and survival to over time. So just a little bit on my progress. I've got about 100 snakes captured since I started in June. And we found out that 5 out of 13 of those locations have RO and 7 have clinical representation of SFD. So I just call it clinical representation because I'm seeing the symptoms of SFD, but I haven't confirmed that molecularly yet. And at my mark recapture sites, you can see the one that's really hard to get snakes at with high RO prevalence. I've only gotten 12 total captures with 1 recapture, or 11 total captures with 1 recapture. Medium RO prevalence, 49 captures and 5 recaps. So pretty decent population sizes. And then low prevalence, 51 captures and 12 recaptures. And I'm just going to end with some cool pictures of pygmies and then kind of what you can do. So I just got a new camera, so I wanted to show these off. Especially like, look at that rattle. And if you see the paint dot, I paint their cloaca to get a better idea of, I use squeeze boxes to measure their SVL. Just because when they're in the tube, they can get kinked up, so it's a more accurate measurement, especially with trying to calculate growth rate, that's important. But I also don't want to disturb them. If I'm going back to that site and I see the animal I tagged yesterday, I just want to leave it alone. So you might see, this is another one that has a little paint dot. This is a young of the year female that I caught last week. This is one of the young Everglades pygmies that I caught. So they're really pretty. This one got a blue dot. And this is the biggest one I've seen, 175 grams in the Everglades. You can see, like, just based on like my forearm, he's almost 3 feet, which is insane, and 175 grams, which is just, yeah, that's pretty crazy. My field tech thought it was an eastern diamondback. These are 2 of the 6 that I talked about that we found dead in situ last year. But if you see something like this, if you see a dead pygmy rattlesnake, you can email me. I have my email at the end. Or you can contact the herp disease alert system, which I should have put the QR code for this on there. But this fact sheet, this QR code will take you to PARC's website where the fact sheet is. And then if you just go to PARC's website, disease task team, then you can find the herp disease alert system. Also, if you just Google herp disease alert system, it'll come up. So if you see a dead or dying amphibian or reptile, contact us and we can kind of get a biologist or state representative out there. And then this is just a QR code for my Twitter. I post a lot of pygmy pictures on there. But yeah, if you see any roadkill specimens that have worms crawling out of their mouth, please email me. And just wanted to say thank you to everyone who has funded the project so far. And then there's my email. But yeah, any questions? That's all I got for you. Yes, question.
You said you found these in captive snakes. Were the snakes captive bred, field collected, and then brought into the hobby?
Yeah, so those 3 specifically were likely collected and then sold, just because one of the garter snakes had a stubby tail, so I'm guessing that it was...
Adults are usually.
Yeah, so they were young adults, but I'm guessing they were collected. I did obviously try to reach out to the person that sold them, but to no prevail, yes. But and then the Nerodia, the one found in Michigan, I don't know that many people that captive breed Nerodia, so maybe it was a wild caught as well. But in Florida, it is legal to take snakes from the wild, keep them in captivity, and then after a certain amount of time, they're considered captive, and then you can sell them. So snakes caught from the wild in Florida are sold nationally and internationally, which could be a cause for spreading this parasite, especially if people, release their snake in Michigan or something. Yeah, did you have a question?
Yes.
Yeah.
RO, where is it originally indigenous to?
Oh, I didn't even mention that. That's a great question. So RO is native to Southeast Asia and likely Africa as well. Mm-hmm. We don't really know much about it in its native range. Yeah. Yeah.
When you're identifying them, how long does that marker stay on them?
Until they shed. Yeah, so it's usually just a few weeks. But I've seen it up to a month. And I only, at the mark recapture sites, I only sample those animals again if it's been a month or beyond. Yeah.
I have a really bizarre question.
Okay, I'm ready for it.
If we find a snake
Yeah.
for you, and you come out and collect it,
Mm-hmm.
can we have the snake back to use as a teaching, just
Yeah.
taxidermy? Because it's gonna be dead if we're calling you.
Yeah.
If it's in invasive.
Yes, I have given people snakes that don't go, so I just take some genetic samples for the museum and then I'd use a picture to voucher the snake for the museum. But I've skinned eastern diamondbacks for people because they want the hide and they collected it. And also with protected species too, some people want the hides of those. Those are a little bit trickier, but yeah, no, whatever snake you give to me, you can have. Yeah, that's not a bizarre question. I honestly get some people will skin the animal, take its rattle, and then give me the rest, which is I didn't know they were doing that, and they did that, and I got the animal, and I was like, what happened? But yeah, hides are used a lot in education. I think that's important, so of course, yeah. You're thinking about it?
I'm thinking about how to phrase it.
Okay, yeah, take your time.
She's a total snake.
Oh, so me too.
I would have never guessed.
Yeah, I mean, venomous snakes are definitely more of my favorite, but I like all snakes.
For snake fungal disease,
Yeah.
is it only on the surface of the skin?
No.
Okay.
Yeah, the hyphae can like grow down into the body cavity. So you can, and often in experimental infections, they'll actually inject the fungus into the body, which, I don't know, maybe doesn't give the most accurate results as to like ecological relevance, but it does get into the body cavity and can be in other organ systems beyond the skin.
And would it be possible to develop almost a vaccine sort of preventative for these snake populations?
Maybe. They've done it with like hellbenders, for instance. They have like, they put a device in them that has an extended release for medication for chytridiomycosis, I believe. But no, I don't think anyone's really tested that for snakes.
I'm just trying to think of preventative measures.
Right. I think the biggest one is, cleaning gear and stuff, but we really don't know. Like one of the things that I want to do in the next year is have my lab technician will swab her boots after each survey to see how much of a pathogen she's actually picking up, especially at sites where we're like, this pygmy has fungus, and then she just like walks around a bunch, and we'll swab it after to see how much pathogen she picks up, but we just really don't know how much influence humans have in spreading it, and whether that differs with the different strains. There's just a lot of unknowns, yeah. Right. And that's oftentimes what this is compared to in the literature. Yeah.
Okay.
Yeah, because I mean, it's definitely worse in colder environments and where snakes are going to go and hide in like groups and stuff. Like when the temperatures get really low in Habronacula, timber rattlesnakes and Massasaugas, they get it really bad. Mm-hmm. Yeah.
What does the snake fungus, is it something that we can see with the naked eye?
Not really. I don't have any pictures of the hyphae up here, but you can see it under a microscope. A lot of times you can't actually, I don't think ever, you can't grossly visualize the hyphae of the fungus, but you can typically see like discolored scaled or like lifted scales, and you'll peek under those lifted scales and you'll see the crust underneath.
No, I meant
Oh.
in nature. So if we're
Oh.
walking outside,
No.
is it the fungus that we can see? And
No, unfortunately not. That would make it easier for sure. Yeah. Any other questions?
Thank you, Jenna.
Yeah, you're welcome. I'll stick around if anyone has any other questions. Who's got the pentastomiasis?